Assalamu alaikum and good day, ladies and gentlemen. Thank you very much for joining us today. I'm Hazima Mozam and I welcome you to an online certificate course organized by Comstech in collaboration with International Center for Chemical and Biological Sciences, University of Karachi, Pakistan. The title of this course is Online Certificate Course on Optimum Use of Analytical and Preparative HPLC. HPLC is High Performance Liquid Chromatography Technique. And with this, I thank you all for joining us today. And now I would like to invite Dr. Atiyatul Wahab. She is with us as the technical focal person for this training course. Over to you, Dr. Atiya.
Thank you so much, Hazima. So as Hazima has mentioned, that HPLC is used in a variety of industrial and scientific applications, such as pharmaceuticals, environmental, forensic, and chemical sciences. This is the two-day online certificate course, and which have lectures and also the training sessions. And we have three very distinguished speakers, Dr. Shakil Ahmed, Dr. Yan Wang, and they are from ICCBS, and Dr. Jean Fal Huang from Hunan Academy of Chinese Medicine. Now I would request Mr. Aziz for the recitation of Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان والأرض وضعها للأنام فيها فاكهة والنخل ذات الأكمام والحب ذو العصف والريحان فبأي آلاء ربكما تكذبان صدق الله العظيم.
Jazakallah, Aziz. And now I would like to request our beloved teacher and the Coordinator General of Comstech, Professor Dr. Muhammad Iqbal Chaudhry, for the introductory remarks.
Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. Good morning to the west of Pakistan and good afternoon in the east of Pakistan. We are in different time zones. We all gathered together here today for a very important workshop, yet another important event for learning, for which I would like to first, at the outset, grateful to my team both in Islamabad, Comstech, and also in ICCBS, International Center for Chemical and Biological Sciences, Hazima and her team, and Dr. Atiyatul Wahab and her team for putting together a very good program, and also very distinguished speakers. You know, I know them personally well, that they are the real practitioner of this extremely important technique. You see, we need to understand that it's always possible to procure equipments. And I have seen this in my own experiences, my visits to West Africa, Central Asia, that people procure expensive piece of equipment and they then place them in different laboratories. But the effective use of technology is an extremely important skill. Without the capacity to use these equipments properly and effectively, you may waste this huge investment made in the procurement of such equipments. At the same time, it also lead to, if they are not effectively used, the results could really be misleading and if, to some extent false. And it is very, very important that we know the technology from the bottom, understand them pretty well, and know how to use these technologies effectively. And that includes all aspects of it, designing of experiment, using right material, right solvents, uh, right interpretation of the results, and at the end of the day, the product which you obtain from such isolation technique should really be effectively pure, which can be used for different purposes. HPLC has, as mentioned by Professor Dr. Atiyat al-Wahab, is an extremely important equipment. You really find them everywhere in the world. Hardly any laboratory, biological sciences lab or chemistry lab or food science lab where HPLC is not present. It is an equipment which you find them in academic setup, in an industrial setup, in government laboratories. And it is often a technique which is most extensively used. So it is used for quantitative purposes, it is used for qualitative purposes. You use it for the detection of contaminants, for quality assurance. You use it for the isolation of compounds from plants or other sources. You use this HPLC for protein isolation of different kinds. So there are huge range of applications of HPLC. And I'm not really aware of any other technique which is so widely used as HPLC is used. So if it is one important technique in which chemists, biochemists, food scientists, environmental scientists, industrial scientists would like to develop their capacity is HPLC. I remember that among our colleagues, people who knew HPLC were able to get jobs in industries pretty well. So it is a tool which is effectively required in every setup, and your effort to learn this technique is extremely important. This series of workshops, which we have organized on the forum of comstech, is meant to develop various skills. And this is one of many events which comstech is organizing. Comstech is a very happening place. You really find activities taking place literally every day. And this workshop certainly add into your knowledge also. So please do benefit from the presentation which some very competent colleagues would make. Use them as a basic set of skills in your own practical experiments. And if you feel that there are still areas in which you need to troubleshoot and develop further capacity, do send your feedback to comstech. And certainly there is always a possibility of organizing such events again in a more elaborate manner on certain aspect of it. So have a great joy of learning, and I welcome you to this forum, which is already over 100 participants, and let you interact with the resource person. Thank you very much, and Assalamu alaikum wa rahmatullahi wa barakatuh.
Thank you so much, sir. We have actually more than 500 registration, and there are in Zoom we can allow only 100 participants. The other participants are joining through YouTube. So now I would like to generate a poll here first. And I would like to request all the participants that in a few seconds you will get a poll and you can answer this poll. And we would like to know how much knowledge you have initially about the HPLC. Can you please hold one? Okay, share हो गया? You can see? Okay, so आप इसको fill करें. You can fill this up also. Yeah. It's about 30 seconds or 1 minute. We will give 1 minute. I think in a minute time we will turn off the poll and we'll see the result. And kindly share result with everyone. We have our IT, very competent IT staff with us who is helping us in organizing this workshop, online workshop. Can you please share the result? Result to everyone. Okay, all right. So the first question was analytical HPLC part of your field of study? So 78% people have said that yes. And the second was, how would you rate your level of understanding of HPLC? So 39% said actually basic, and 53 have intermediate. 5% is advanced, and 3% are the expert. And the third question was what level of experience you have regarding separation, identification, and quantification of components of a mixture. So 41% said basic, and 47% intermediate, and 13% are advanced. So actually, we would like to see this answer from you, so that we can actually have the discussion accordingly. So now we know that most of our, about 50% of our participants have the basic knowledge and understanding, and about 50% have intermediate, and advanced are very, very less. So basically, our the first speaker, Dr. Shakil, will now start, yeah. Start from here, and I would like to introduce Dr. Shakil. He is a very renowned chemist in charge Industrial Analytical Center at HEG Research Institute of Chemistry. And Dr. Shakil has years of experience working on analytical HPLC, and he is the one who have done lots of troubleshooting during his career. And he has very much experience in the field of forensic sciences, environmental protection, pharmaceutical industry, and there are a lot more about him. So I would request Dr. Shakil, kindly come here and start your presentation.
Okay, thank you very much, and dear participants. As pool shows, the basic knowledge of HPLC has about everybody. So we are starting this presentation because this is the online learning HPLC course. So we will discuss everything which we use in analytical HPLC in this presentation. And tomorrow's presentation, I will explain how to operate the analytical HPLC. That is a practical demonstration. Today, we try to brief you everything regarding the analytical HPLC, and during the use of the analytical HPLC, what type of problem you are facing. Before starting analytical HPLC, we will start with the chromatography. This is the basic terms used for HPLC. HPLC is a kind of chromatography. What is the chromatography? Because chromatography is the technique which we use for separation of individual components, that is physically and chemically from the mixture. Chromatography word derived from the Greek word. This means chroma. Chroma is used for color, and graphy means study. So the HPLC is the okay. 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 So now you can you're watching this on this presentation that on your right in your right where we are marking. This is the column which we are using for chromatography. In this column, we load or we introduce some sample on top, and this band is showing the separation. There is a different type of separation technique we are usually we use. That is a classical separation technique, and this is a modern separation technique. For classical separation technique, we are using the crystallization, precipitation, extraction, sedimentation, adsorptive filtration, and distillation. And this is the modern technique which we are using for the separation. That is paper chromatography, high performance liquid chromatography, gas chromatography, thin layer chromatography, liquid chromatography connected with mass detector, gas chromatography detected with mass detector, and HPLC. It is the high performance thin layer chromatography. Okay, the chromatography technique divided in two big groups. One group is gas chromatography, and another is liquid chromatography. For gas chromatography and liquid chromatography, both are using in analytical separation. For pharmaceutical industry, for environmental industry, for any kind of space. The difference in between these two, for gas chromatography, we have the analytes which we are analyzing, which is thermally stable and volatile. For liquid chromatography, we are using the substance which need to be analyzed 100% soluble in mobile phase. HPLC or liquid chromatography divide into TLC, which is basic chromatographic technique, and HPLC, high performance liquid chromatography, paper chromatography, which is also used. There is another technique we are using nowadays, UPLC. This is high pressure or high performance liquid chromatography. And solid phase extraction technique is also using in our analytical separation and flash chromatography. This is the mode of liquid chromatography which we are using for separation. Here you can see this is the TLC card or TLC strips. This is the spot which we are showing here. This is the sample apply on TLC, and this whole TLC is maybe from made by the glass or alumina, and the absorbance is maybe silica or alumina or anything. And at bottom you can see this is the liquid. It is solvent or mobile phase at time 0. This this sample is on the bottom or at the place where we apply. And in second TLC, you can see the different spots. This is different spots is showing the separation on TLC. So after applying time and running the solvent, this is the separate. This is the basic for every chromatography. When we apply any kind of substance absorbance and the mobile phase, according to the nature of the substance, we need to be chromatographed. It is separated into the different according to their polarity and nature of the component. Okay. And another kind is the paper chromatography, which is not so much popular, but still the people are using paper chromatography. Okay, then another thing is more important. This is column chromatography. Okay, column chromatography normally, which in lab we are using this solid phase extraction. This solid phase extraction, there is some cartridges column is available for this purpose. There is a different cartridges columns are available. There is a normal phase cartridges column, reverse phase cartridge
Yes.
Can you hear me?
Please solve the problem with speaker voice.
Everybody is
problem है. देखिए जरा.
okay. So let's start again.
Now
Because
yes.
sorry for this inconvenience. Okay.
Thanks कर दो.
Okay, thank you very much. Let's start. This is the typical example for chromatography or cartridge chromatography. We apply the sample on top of the column, and in first slide you see this is the sample loaded on absorbents. After that, this in second slide you can see the blue, red, and yellow. The first yellow band is collected in the beaker, and in second slide, when mobile phase running, the second band which is here is down, is collected in the reservoir, and after that the third band is on the basis of this. In previous slide, I already shown you TLC is against the gravitational force, and this column is towards the gravitational force. So these all bands are separated and collected through the column or solid phase extraction or semi-preparative HPLC or preparative HPLC, but not analytical HPLC used for the collection of the sample.
Next. नहीं होगा.
Okay, so okay, now second pole. Please show the second pole. Time is 1 minute. Please fill it and submit it. The first question is analysis technique.
What is chromatography? What is chromatography?
What is the chromatography? About 13% answer analytical analysis technique, identification technique, separation technique. 81% says technical separation technique. Okay, every person is right, and this is the chromatography. What is the chromatography? Is the separation technique. Identification through chromatography, yes, you can, but when you have the standard, substandard substance, then you can use the chromatography for the identification. Analysis technique. chromatography, as on the third or second slide, I told you chromatography is a separation technique. Mainly, chromatography used for separation. Identification only possible when you have some standard against the sample. Against the standard, you can identify the component. What is the chromatography? Which is the chromatography techniques? Second question, UV, HPLC or mass. So about every 98% give correct questions, correct answer. Yes, this is HPLC. Among these three, HPLC is the chromatography technique. HPLC stands for high pressure liquid chromatography and liquid chromatography. Actual word is high performance liquid chromatography, but some person is also HPLC stand for high pressure, but the correct answer is high performance liquid chromatography. Okay, let's start with our presentation. Okay, high pressure, high performance liquid chromatography is the type of chromatography. It is a column chromatography. Is a type of column chromatography. It is used for separate, identify, and quantify the components from the mixture. The cartridge is we are using cartridge or column is packed with the stationary phase. Can you change this slide because this okay. A liquid mobile phase passes through the stationary phase, that is a column, and a dissolved analytes is injected. I already mentioned that the analyte is the requirement of that HPLC analyte 100% solubilize in the mobile phase. Then you inject it or subject it on the column along with the mobile phase. The mixture of analytes separate into individual components or band in the form of peak. The band separate, which already shown you through the column, and the peak is electronically signals shown by the computer. The graphical presentation generated a chromatogram shown peak of analytes present in the mixture. Peaks are then quantified. This is the type of chromatography on the basis of separation and on the basis of eluent. On the basis of separation, we are using two kind of the column. One is the normal phase column used in HPLC. Another one is reverse phase column used on analytical HPLC. On the basis of eluent, two kind of eluent mode we are using. One is isocratic mode, another is gradient mode. Okay, I will explain the maybe many people knows about this isocratic more detail in the next slide we will discuss. This is in isocratic mode, we are using single mobile phase, not single solvent, single mobile phase. In gradient, is more than one mobile phase we use. Mode of separation is a normal phase chromatography. Stationary phase in normal phase chromatography is the polar, which is hydrophilic, and mobile phase is non-polar, hydrophobic. Means the normal phase column, normally we are using normal phase column, silica column, and mobile phase, normally we are using hexane or other non-polar solvent. This is the most common technique we are using for analytical HPLC, that is reverse phase chromatography. Here, in the reverse phase chromatography, the stationary phase is non-polar and non-polar hydrophobic, and mobile phase is polar.
There are some questions also coming up. So can we interrupt?
Sure.
More interactive? So there is a question from Gil Bibang, and he said that can we quantify the components with just area peak?
Yes, area peak.
Okay. When we are using analytical HPLC, or if we have one thing I already discussed on HPLC, we can quantify. Requirement is only one. We have to have a standard against the sample. If we have the standard peak area of the standard, and we have the peak area of the sample, with the comparison of standard, we can quantify. With the comparison of the standard, please note down, with the HPLC cannot only on the basis of peak area alone cannot quantify. Is there any more questions? Okay. It is very good. Somebody is interacting with this and listening carefully about this presentation. If any more question, because this is the online course and the audience is not in front of me. So if there is any question, just please feel free, ask. And if I know, I will answer it. And hopefully you will learn more about this. One more question. So Dr. Atiaz is asking about one more question.
This is the question from another participant's name, Bissy Flora Davis, and she's asking about, I think she or he, what is the minimum concentration and volume to use to separate crude extract from microbial metabolic compounds, which are usually obtained in very minimal amount?
Okay, there is a very tricky question, but I would like to answer, because this is a practical approach. Because for from the microbial, very minimum quantity is obtained. At that time, you have very smart. You just because metabolites is on an analytical HPLC is a very difficult to isolate the compound. For this purpose, we have the fraction collector type. One fraction collector should be attached with the analytical HPLC, and that fraction collector has microplate 96 microplates, 96 well microplates, in 96 well microplates, we are usually collecting the small quantity of the metabolites, which is obtained from the microbial. Any more question, Dr. Tia? Okay. Okay, which is a good during presentation. If we discussing everything, then it may be more informative and interactive presentation. Then because it is a basic. If you are not asking anything, only just we go through the slides and everything. Now you can see the here I already discussed about the normal phase and difference between the reverse phase, and this is the interaction between polar bond and non-polar bond. We have a more affinity than polar non-polar bond. Reverse phase chromatography is a more common use as a drug are usually hydrophobic. In pharmaceutical industry, they are mostly use this chromatography. Because reverse phase or some the RP18 or some the RP8 column they are using. This is HPLC is not reverse phase. Only the column is reverse phase. So so keep in your mind, this is reverse phase means we change the phase of the column or material in the column which column was packed is the reverse phase. Principle of separation. This is absorption chromatography. In the absorption chromatography, solute molecule bond directly to the surface of the stationary phase. The compound which has more affinity towards the mobile phase elute first. The component which is has less affinity towards the stationary phase elute later. No compound has same affinity towards the mobile phase and stationary phase. When we are separating any compound or mixture of the compound, at least in two components, in physical properties should be changed. On the basis of physical change of the physical property, we are separating these compounds. Another kind of the chromatography, it is all ion exchange chromatography or ion chromatography. This chromatography is used in the environmental, mostly environmental and protein chemistry. Ion exchange chromatography allow the separation of the ions and on the basis of polar molecule base.
One more question, and this is a question from Hafiza Sadia Mutalib, and she is asking why use of more than one mobile phase in gradient analysis.
Okay, is good, Sadia. This is a good question. I already, in my previous slide, I mentioned that in isocratic mode, we are using one mobile phase. Is not necessary. One mobile phase is the combination of only one solvent. It may be combination of two solvent. It may be combination of three solvent or more than two. But in gradient mode, we are changing the concentration or polarity of the mobile phase with respect to time. So with respect to time, when we increase the polarity, we remove the impurity which is interacting with our target molecules. For that purpose, we are using the gradient mobile phase. It is more for R&D work. In R&D work, never any R&D chemist use isocratic system. That once we develop the system, in gradient mode, then we convert it into the isocratic mode, because gradient mode is removing the first impurity or first peaks from the column, and after in gradient mode you can obtain the peak in sharp position, because you are changing the mobile phase with respect to time. And another thing is for the better resolutions. I think this is more interactive session. So people are asking questions. It means they are listening this lecture carefully.
So another question. This is the question from Raji Golaji, and the question is, please explain why are we changing the concentration of the mobile phase?
This is the same question, which already answer it for better understanding, the better resolution, and get the better peak. And but this question and the previous question is the same. Okay, so the retention is base of which ion exchange chromatography. I'm explaining this one. Allow the separation of ion and polar molecule based on their charges. Mainly in protein chemistry, you know there is a several protein has a different charges. In ion chromatography, you know cation or anion separation of chloride, bromide, nitrate, sulfate through charges. So this ion exchange chromatography are using for this purpose. It can be used for almost any kind of charged molecule, including large protein, small nucleotides, and amino acid. This is a nitrogen containing positive charge. Retention is based on the attraction between the solute ions and the charge side bond and stationary phase. Ions of the same charge are exchanged. The use of resin, resin is the stationary phase in ion exchange chromatography, and solid phase is a need for covalently attach anion or cation on it. So these all are type of the HPLC which we are using. Just we are changing some parameters. Okay. This is ion pair chromatography. In ion pair chromatography, it is a form of chromatography in which ion in the solution can be paired and neutralized on the separation and ion pair and reverse phase column. Okay, ion pair, actually, if some audience is listening about and they are using HPLC, sometime when you are making mobile phase, you use some ion pair regions. These ion pair regions is the purpose of that ion pair region is if some two peak is overlapping and difficult to separate through pure mobile phase, at that time we are using that ion pair region. And ion pair region is usually anionic compound and contain a hydrocarbon chain that impact a content of hydrophobicity, so that the ion pair can be retained on the reverse phase column. This is most commonly ion pair region which we are using is hexane sulfonic acid, if you aware about this. So this is a more commonly ion pair region we are using in reverse phase chromatography when two peaks are not separate. Gel permeation chromatography, this is another kind of the chromatography HPLC technique we are using, but this HPLC technique used mainly for determining the molecular size of polymer. Okay, the type of the chromatography is like interactive between the stationary phase and solute. This liquid or gaseous phase through the porous gel, which separate the molecule according to its size. The poses are normally small and excluded large molecules but allow similar molecule to enter the gel and causing this flow through large volume. The cause the large molecule to pass to the column of faster rate than the smaller. This gel chromatography is using mainly the polymer chemist use this chromatography. Is there any question, Dr. Atia?
Okay, there is one question. This is the question from Wasim Akhtar. Is there any difference of phases from plant and animal samples?
Okay, then we are discussing here the HPLC and use of the HPLC. But your question is more related with the sample preparation. Yes, the sample preparation is the art. Sample preparation is not easy. If we have a plant extract or plant material, sample preparation and cleaning procedure is different. When we have animal tissues, cells, then sample preparation and cleaning stages are different. So once you make a sample ready for inject into the HPLC, at that time you can use that HPLC, but this is more application side. Any more questions? So any more polls? Dr. Okay. Okay, let's go forward. This is the affinity chromatography. Affinity chromatography is also used for chromatography. This is most selective type of chromatography employed to utilize the specific interaction between one kind of the solute molecule and second molecule that immobilize on the stationary phase. The example of affinity chromatography is the chromatography where one technique which we know is that aflatoxin or some other kind of special kind of the material is we subjected on affinity chromatography. That is made by some kind of the protein, one antibody, and then that antibody is retained all aflatoxin on the surface. We clean it, the sample, we clean the column through one solvent, and after that we immobilize another which is bind with the antibodies, and we purify with the help of the affinity chromatography. For example, when we solute contain a mixture of here you can see when solute containing a mixture protein are passed by the molecules, only the specific protein is reacted to the antibodies and binding it on a stationary phase. This protein is larger later on extracted by the changing of their ionic strength, the solvent, pH, and so on. This is very tricky chromatography. Is a chiral chromatography. This chromatography is used only for separation of enantiomers, stereoisomers, which is not which is identical. our mirror image is not possible to separate through a chiral mode. So for this purpose, in this case, the enantiomers, these have no chemical and physical difference apart from this being three-dimensional mirror images. Conventional chromatography means our normal phase, the reverse chromatography, or other separation techniques are incapable of separation them. To enable chiral separation to take place, either the mobile phase or stationary phase most themselves made chiral. If the mobile phase is chiral or stationary phase of chiral, if anyone is chiral, so one, if we have a mixture of two components, one component will interact with the stationary phase, or the mobile phase is more interacted with stationary phase or more interacted with the mobile phase. At that time, we separated the enantiomers or stereoisomers. Okay, this is the isocratic. This is the technique which already I mentioned before, elution technique, which based on isocratic mode, a separation in which the mobile phase composition remain constant throughout the procedure in the isocratic elution. Here, if we have a 4-pump quaternary gradient pump, just for the understanding, if we have in one pump we have a methanol, and second pump we have a acetonitrile, third pump we have water. If we are using all three pumps one time, and throughout the analysis we use same composition of the solvent, is called the isocratic elution. Okay. In isocratic elution, peak with, what is the disadvantage of isocratic elution? The isocratic elution peak with the increase of retention time. Isocratic elution increase the retention time linearity with the number of theoretical plate, which I will explain this theoretical plate later on. This lead to be a disadvantage that later eluting peak get very flat and broad. This is the main disadvantage of isocratic mode. The peak will, because it takes longer time and peak is flat. Best for sample preparation, often used in quality control application that support and are in a close proximity to the manufacturing process. Once we set the method on a gradient, we convert it on isocratic mode. Gradient mode, this is the, we discussed already, this is more common elution in HPLC, a separation in which the molecule phase, mobile phase composition is changed during the separation in decreasing a gradient. But if you are doing the column chromatography, you are also changing the polarity of the solvent with respect to the time. Gradient elution decrease the retention of the later eluting component. Decrease the here you can just please try to understand about this. The gradient elution decrease the retention of the later eluting component, so that they are elute faster, giving and narrow peak. This means the sharp peak. This also improves the peak shape and the peak height. Best for the analysis of the complex sample. If you have more than 5-6 compound in one sample, is better. We use gradient mode. Often used in the method development for unknown mixture. linear gradient are most popular. We have another poll, so please fill it and submit. Please keep in your mind, only 1 minute you have, just fill it and submit. Okay. Well, first question, which is the correct statement? I think so. Most of the people answer correct, about 73%. Okay, and isocritic mobile phase considered as pure solvent. Yes, for HPLC, always we use pure solvent, but this is not right answer. Pure solvent, mixture of solvent, or in respect to the ratio. This is the correct answer. Two pure solvent in two different reservoir. Yes, again, pure solvent is used in HPLC. So the correct answer is the mixture of solvent in a specific ratio. Gradient system considered as the two pure solvent in two different reservoir. Solvent composition change gradually throughout the system, throughout the analysis. Yes, this is the correct answer, or most of the people answer correct. So this presentation is more interactive just because of you. You are attending the questions, asking questions during the presentation. So if there are any question?
From Radayati, and he asked that sir, guide us on how biotechnologists use HPLCs to separate and identify different foods.
For protein, I already presented one slide. Then ion exchange chromatography can be used for identification of protein, but a small molecule, not macromolecules. And at that time you have a specific column of HPLC and a special mobile phase. Normally we are using C4 column for this purpose. C4 again is a reverse phase type, but for carbon chain is derivatized with C4. Okay, again now our actual target which we want to demonstrate you that of type of HPLC we are using. Here we are using in laboratory, we are using this kind of HPLC, that is the analytical HPLC or UPLC. Here we are using semi-preparative HPLC, which my colleague Dr. Huang will explain about this in detail. And this is most common preparative HPLC used in industry for separation of for pharmaceutical raw material and many more things. In analytical HPLC, normally we are using only for quantification and qualitative work or identification as well. This is the typical diagram. This is the solvent reservoir, and this is a pump which we are using in HPLC. Maybe is a isocratic pump or gradient pump, any kind of pump which is placed after the solvent system. After that, after the pump, we are using here the sample injection mode, which is maybe gradient, or which we normally called Rodine. This is the column packed with the stationary phase. Depend again which kind of the stationary phase in it is depend on nature of your work. Then again, after eluting from the column, the substance goes into the detector, and again this detector is according to your application. Which kind of detector you want to use here. We will explain about the detector and the choice of the detector, how you can select the detector for according to your application. Not always UV detector or PDA detector are using for one kind of the analysis. It's a different kind of analysis, different disciplines people are listening. So different applications can be demonstrate the different detector here. Then this is computer data system, which the detector is response. This is electronically response of this peak, and in HPLC normally these kind of peaks are appear, and this peak will show us their peak height, area, and everything. We'll explain it about this later on. Okay, solvent, which already mentioned you. This is which we already mentioned you. Solvent, mobile phase, reservoir, isocratic mode, gradient mode, which you already knows about, and Paul is also showing you knows about the gradient and isocratic mode. Solvent polarity is very important to choose the for the solvent in during the analysis. If you see, there is an increasing the polarity from downward to upward. This is hexane is less polar, where water is more polar, acetic acid is more polar. Here you can see the increasing the eluting power, increasing eluting power. Hexane is eluting less eluting power, while methanol has more eluting power. I think so. This terminology is everybody understand about what is the eluting power, what is the polarity. So it's almost, if you can see, water and methanol eluting more, is more polar and more eluting power. So normally we are using methanol water mixture is about more than 50-60% analysis is depend on water and methanol. Rest acetonitrile. If we are using 3 solvent, acetonitrile, methanol, water, this is more commonly used solvent in HPLC system. Here, one thing is important here: how many type of HPLC pump we are using. There is a 3 kind of HPLC pump. Most common HPLC pump which nowadays we are using is reciprocating piston pump. This reciprocating piston pump we are using nowadays in our HPLC, and the syringe pump and the constant pressure pump. One by one, I will explain where these pumps are used and how they are working. Reciprocating piston pump, which we are using in HPLC. It is widely used pump, considered as a small, consists of a small motor-driven piston which move rapidly back and forth in a hydraulic chamber that may vary from 35 to 400 microliter in volume. Just to see 35 to 400 microliter volume they consider. On backstroke, the separation column involved is closed and the piston pulse is a solvent from the mobile phase reservoir. On forward stroke, this is all phenomena. How the HPLC pump will work. When the forward stroke, the pump push solvent out from the column from the reservoir, and wide range of the flow rate can be obtained. Alternative, the piston stroke volume during each cycle or by alternating store efficiency. These are 2 kind of things happening at one time. Syringe pump. These are most suitable small bore column because this pump deliver only a finite volume of the mobile phase before it has a refill. These pumps has a volume between 250 to 500 ml. So used for large scale. The pump operates by motorized screw that deliver mobile phase to the column at the constant rate. The rate of solvent delivery is controlled by changing of voltage on a motor. Third kind of pump is the constant pressure pump, which we are using here. This type of pump, the mobile phase is driven through the column with use of pressure from the gas cylinder. A low pressure gas source is needed as generated high liquid pressure. The valving arrangement allows the rapid refill of the solvent chamber, whose capacity is about 70 ml. The provided constant phase is flow rate. Okay, here if you can see, there is a injector of HPLC. You just understand how the injector of HPLCs work. These are 2 kind of injector. One kind of injector is the manual injector. Another kind of injector is the auto injector. If you can see this knob, this is the knob is load, and load position, this is the inject position. In load position, you can see this is the pump 2, 1. This is not connected here, and here you can see this all are connected. When we change from load position to inject position, now our sample goes into the column, and then here you can see the loop, column, loop, then column, and then detector. So when knob in load position, remains injected sample into the loop. When loop is changed from the load position to inject position. The injected volume is subjected on a column. At that time, separation. If you have a manual HPLC and you have a loop and you injected a sample on a loop at load position and you are waiting for separation of the compound, during these time when your knob is not changed from load to inject position, your separation of is not subjected on a column. So HPLC, there's no analytical work is going on, which already I described everything about this manual injector. And one thing, if you have a less work, then you can use manual injector. But if you have constant work and accurate, this is there is another problem for injector volume. When you inject any volume in the loop, if you change the volume through syringe, there is some error is occur here, because if you have a 20 microliter loop and you are injecting 10 microliter, means you are injecting 10 microliter through the syringe. If you are expert in the HPLC, you always try to inject volume in a fixed mode. Means if you have a 20 microliter loop is here, so you just inject full loop, not partial loop. Partial loop always creates some problem. But here, the advantage of the auto injector here, because this is the automatic system, which you give the command in the software pick the accurate volume through injector. So you give the peak area reproducibility better than a manual injector if you are using partial loop. If you are using full loop, okay, yes, you can get the more precise result here. And auto injector advantage, you can multiple inject with respect to the time, and if you have hundreds of the sample, you can place in auto sampler and make a batch file and start run. So is a more convenient for automatic HPLCs. Here already we discussed, this is the silica is attached with this C18. This is silica derivatized here again with C8. For this type of the column, we call C18 column. This one is C8 column. This is the normal silica column, which we are using for a normal phase chromatography. Sometimes we are using wide volume when this particles packed with the in column, the solvent is surrounded with the this kind of this type of stationary phase is called wide volume. Shamshad, kindly show us the poles now. One minute is Okay, HPLC process flow equation. There is a maybe there is a confusions. Okay, reservoir, pump, sample injector, column, and detector chromogram. Reservoir, sample injector, then pump. Maybe I already shown you the when we have a mobile phase. After the mobile phase, there is a pump. After pump, the solvent. The reservoir process flow is always reservoir, pump, sample injector, column, and detector. This is the right answer, because the reservoir, sample injector, if we have a reservoir, who will inject the sample? Pump will inject, not reservoir. Okay, second one is solvent delivery system in HPLC carried out by means of solvent delivery system is pump. Right answer is the pump. Column packing usually composed of C8, silica, reverse phase, C8, C18 reservoir. Normally, C8 for normal phase. For normal phase, I already show you the normal phase is without any derivatization. Means in normal phase is silica, only pure silica. Silica for reservoir, for reverse phase, silica is not reverse phase. If we have a hydroxyl group of silica dioxide is derivatized with C8 or C18, then we are using the reverse, when it's converted into the reverse phase. C8, C18 reverse phase is the right answer, is about 69% audience give the right answer. So let's go. Another question? Okay. What is the wide volume? Okay, volume is again 50-50%. Mobile phase required for saturation of the column. Volume of mobile phase required for eluent of the peak. The mobile phase required for eluent of the peak is the wide volume. Okay. Okay, the most important of HPLC is choice of detector. Here, some common detector which we are using in HPLC, which is showing is more than that, but some common detectors are here. One detector is the UV detector, another is PDA detector. UV detector and PDA detector both are using. The UV detector is using on a fixed wavelength. On PDA detector is using on a variable wavelength, on a scan mode, and 3-dimension. RI detector is the refractive index detector, which we are using in HPLC. ELSD detector, evaporating light scattering detector, and the fluorescence detector, ECD detector, they are using conductivity detector. This is depend on the application. If we have a chromophore, we use UV or PDA detector. If we don't have any chromophore, like sugar molecule, we use RI detector. If we want to use ELSD detector, evaporating light scattering detector, this also we can be used for without a chromophore molecule can be analyzed through this. The difference is the RI detector is work only on isocratic mode. RI detector never use in gradient mode. ELSD detector for UV inactive compound also we can be analyzed on through ELSD detector. This ELSD detector can be used in a gradient mode. Fluorescence detector is a requirement. We have to have excitation or emission for if we have a component have excitation or emission, we use fluorescence detector. For example, for fluorescence detector, we are analyzing the aflatoxin or mycotoxin through fluorescence detector. ECD detector is using for is a specific use. Neurotransmitters can be analyzed through dopamine, and others can be analyzed through ECD detector. Conductivity detector, this detector can be used for analysis of ions. Suppose chloride, bromide, nitrite, sulfate, ions can be analyzed through this conductivity detector. Is also called, in generally people are saying for ion chromatography. Is also kind of the HPLC. This is the flow sheet diagram of UV detector, light, monochromator, then computer signal appear here. So this is the typical diagram of the UVA or PDA detector. Already mentioned about RI detector. In RI detector, there is a reference cell and mirror, and flow cell is balanced. After that, after when flow cell is balanced, then this will work as on the basis of their refractive index. That is the reason we are, if refractive index is changed, the peak will appear. This is the reason we are not using the gradient solvent system in this. And always keep in your mind, and is a practice, once you analyze you are using this detector, you just make a mobile face once and close it. If it is the concentration of the mobile face is changed during the operation of the HPLC, if any change is appear as in resulting in the form of peak. This is the evaporating light scattering detector. Here, this nebulizer are using. There is a space chamber. There's a the molecule, the mobile, some compound with the mobile phase nebulized here, and then scattered the light, and then detector will detect the compound. Fluorescence detector is very specific, which already mentioned. If there is a UV detector, fluorescence is much less common than UV detector. Application I already mentioned you, mostly used for aflatoxin analysis or mycotoxin analysis. ECD detector, which already mentioned you, for the analysis of dopamine, serotonin, noradrenaline. And conductivity detector, mention you about this is some advantages of the detector and some disadvantages. UV detector advantage, if we have a chromophore, we can use. If we this is a non-specific complex is disadvantage is non-specific complex sample absorption bandwidth. Dyed array works for all wavelength, because if we can analyze, if we are analyzing 190 to 400, we can analyze one time in UV detector. We fix the wavelength or tune the wavelength, but in dyed array detector, we are using in scan mode. And fluorescence is very specific. Refractive index detector, we already mentioned on the basis of refractive index, good for sugar molecules which don't have any chromophore. Light scattering detector, if any component which is RI on the basis of RI or light scattering, so gradient mode can be used. Here, electrochemical detector for neurotransmitters, mass detector. If we use mass detector with HPLC, it converted into the LCMS, and mass detector is a very, very sensitive technique. If you know the basic HPLC, only detector change, and change means the sensitivity is changing. Nowadays, the residual analysis is using through this detector. I think so. Is detector is all the detector we explained here, and time is running short. So I just stop here, and tomorrow the left of the slide I will explain along with some practical demonstration how we are using the analytical HPLC. So thank you so much for your patience. Thank you so much. Yes, if you any question, please ask. Feel free.
Okay. Thank you so much, Dr. Shakil. Was very interesting and informative presentation. Now I would like to introduce another very renowned chemist, Dr. Jean Fal Huang. He is assistant professor in Hunan Academy of Chinese Medicine, Changsha, Hunan Province, China. He was elected as an excellent young innovator of Changsha City in 2018. At the very young age, his major interest in research are on three topics. Number one is machine learning algorithm development and application in metabolomics, bioinformatics, and pattern recognition. Number two, quality control and new quality estimation method development for traditional Chinese medicine. Number three, comprehensive application of herb resources. So now I would like Professor Jean Fal. Professor Jean Fal, are you here?
Yes, I'm here.
All right, so now it's over to you. Can you please share your slides?
Okay, thank you very much for your introduction. Okay. Hello.
Dr. Jean Fal, can you please turn on the camera as well? We can't see you. We can see your slides, but we cannot see you. Please turn on the camera.
Okay.
This is perfect. Now we can see you, but there is some problem in sharing the slides. Can you please share again?
Okay.
Yeah. Yeah, now it is okay. Yeah, over to you, Dr. Jean Fal.
Okay. Thank you very much for your introduction. And Assalamu alaikum. Hello everyone. This is Jean Fal Huang from Hunan University of Chinese Medicine, and now I'm in Changsha and Hunan Province. So first, warm welcome to join this HPLC workshop, and thanks a lot for International Center for Chemical and Biological to organize such kind of online certificate for HPLC, and also thanks a lot for ICCPS and Professor Iqbal and yours to invite me to give the lectures in this course. Okay, let's begin today's course about the HPLC. Today, I want to introduce the 4 parts of lectures about HPLC. And firstly, is the introduction of HPLC. Yeah, because Professor Shaki have given very detailed introduction of HPLC already. So here we will go very simply. So as we know, HPLC has many advantages such as high speed, high pressures, high efficiency, and high sensitivity and widely application. HPLC is very suitable for efficient separation and analysis of organical and biochemical samples with high boiling point and similar instability compounds. So firstly, is about it is widely application. Normally, more than 16% of organical compounds can be analyzed by HPLC. And here I will give you one example. Now you can see this is the Chinese pharmacopoeia. Normally, Chinese pharmacopoeia, the herbals can be used in China has been recorded, and recorded from 1994 to 2010. Each 5 years, it will update 1 time. And we can see from here, in 1994, about 920 plants has been recorded in Chinese pharmacopoeia, and only 11 liquid chromatography has been used in but in 2010, about the herbals has been recorded in Chinese pharmacopoeia increased to 14,016.4, and we can see how the method used the liquid perform chromatography has been 20,064, and also the percentage of the liquid chromatography be used increased from 1.2 increased to 86.4. So that means most of the food herbals and can be analyzed by using HPLC method. Here, the second advantage of HPLC is high speed. Normally, it's much faster than the classical liquid chromatography. Usually, analysis of a sample in 15 to 13 meters. Some samples, even in 5 minutes, can be complete. So generally, the whole analysis time will less than 1 hour. So it means it has very faster speed. But of course, if you want to get high speed, you must need first one. You need high pressures, because the mobile phaser need very quickly through the chrome, and this is provided by the pump used. So that's normally it will give a very high pressures. In the meantime, if you have too faster analysis speed, the high efficiency is necessary. High efficiency means you can separate many compounds just in one time, And another one is important is high sensitivity. High sensitivity means that sometimes because in the compounds in the herbal, normally the concentration will have a very large difference. Sometimes maybe in the granted, sometimes in the mirror granted. So sometimes you must need high sensitivity for HPLC method. Okay, here also we will give a simple introduction about my compounds of high performance HPLC method. In here, just as Dr. Shaki has been giving us the introduction, here they have something mobile phaser, and then here is a pumper. The pumper will give transfusion the more mobile phasers, and then we will have injection vial, and the injection is used to inject the samples into the mobile phasers. And then another one is column. In a column, we use to separation the compounds, and then after the separation, all compounds will went to the detector. After the detector, they will change to the thinner, electro thinner, just like this, and this will be recorded in our computer. Now this is the HPLC working principle. Okay, now we will introduce the main compounds one by one. The first one is high pressure infusion pump. This is the high pressure infusion pump, which is can drive the mobile phasers and the samples through the separated column and also into the detector system. And this one is here you can see is the injection valve. Now this is normally we call it 6-way valves. And also Dr. Shaki has to show how it works. This part is very important, is column. In column, normally we have 2 types of column, one for analysis tab, and another is for laboratory preparation tab. And here, for the analytical tab, the inner diameter weighs 2 to 6 millimeters, and for the column length, normally is the length will be 10 to 24 centimeters. And for preparation tab, the inner diameters will be 20 to 14 millimeters, and the column length will be 10 to 13 centimeters. Besides that, we also have a positive phaser column and inverse phaser column, and also this is defined. Also, the inverse phasers column has been widely used. Now, this is the separation principle of HPLC column. And here we can see the mobile phasers has been here, and the samples can be injected by the injection valve, and it can be here, because then the mobile phasers will went down like this by the pumper. And the compounds in these samples here will be separated because they have a different retention time and retention ability. So this is the simple separation principle of HPLC column. Okay, last 2 compounds of HPLC method. Here we can see first is the detection system and the data processing system. And these 2 part system work together because they convert the concentration changes of the analysis into an optical and electro thinner. Okay, and here normally we have workstation for this to process this data. Okay, next we will show you 2 common used high performance liquid chromatography. I will show you the video about them.
建华兄，现在你是可以说话的，你是可以说一些话来解释这个。他们说问是不是声音不好使，是不是他们以为这里是应该有解释的，然后听不到。
哦，这个我们they have videos in the voice in the video, but I don't know why when I show it, so you can't hear about them.
Just screen share with audio.
Oh.
There's option of sharing audio.
分享里边说是也可以分享声音，如果这边是听不到声音的，
好的。
你可以解释，也可以通过这个你讲的方式。
好的 ，OK, I will introduce of them by in the video also.
Opening the mic. Okay. Speak. Speak.
建华兄，他们说你要重新分享，分享的时候要把这个就是和声音一起分享。他说是有一个选项，把那个勾选上，声音就可以好使了。
Okay, I will try it. Okay, I will try it. Okay. Yeah. Okay. 我们那个在哪里呢？So can you hear the voice now?
Your mic is open up here.
Dr. Jean Fal, can you please, when you share the slides, can you please check on the share sound?
Okay, I will check them.
Check on the share sound.
Share sound.我们分享了啊。OK，屏幕在哪个地方？
Dr. Jean Fal, when you are sharing the video, then there is an option of share sound. Please check this one.
这里有啊，是吗 ？Is here? OK。这个怎么 how to collect it? Okay.
C and D, which are used to store the mobile phase. Note that the suction filter should be placed below the liquid level and cannot be exposed to air.
Now the voice is clear.
Will be generated and the use of the instrument will be affected. Below the tray is a high-pressure infusion pump. On the left is an online degasser, and on the right is a
now the sound is clear. Can you please play from the beginning? From the part where you want to start?
Okay.
Yes.
This instrument is a high-performance
voice is
liquid
okay now.
chromatograph made by Agilent, USA. Let's first briefly introduce the hardware configuration of this instrument. First, there are 4 liquid storage bottles A, B, C, and D, which are used to store the mobile phase. Note that the suction filter should be placed below the liquid level and cannot be exposed to the air, otherwise air bubbles will be generated and the use of the instrument will be affected.
Dr. Jean Fal,
Below
can
the
you
tray
please
is a high-pressure
get
infusion pump. On the left is an online degasser, and on the right is a pump head. The exhaust valve should be opened to exhaust before starting the machine. And there is the automatic sampling system. On the left are 2 sample trays for placing the samples to be tested, and on the right is a 6-way valve. Then there is a column oven in which a chromatographic column is placed to separate components of different polarities in the sample. At the bottom is a detector, which transmits the signal to the computer to get a chromatogram. Let's take a look at the high-performance liquid chromatograph in Shimadzu, Japan. First, on top of the tray are bottles containing water and organic phases. There is a suction head inside the bottle to suck up the solution. Do not expose it to the air for a long time in the process. And down here is the automatic sampler, where we put the process samples. Here is the position of the 6-way valve. The following 2 parts are online degassing machine. You can turn the knob for degassing. The next part is the column temperature chamber, where our column is located. And the simple way that the liquid works is that we pump the mobile phase and we take our sample to the column. We use the column to separate the substance. Then the substance goes to the detector. The detector sends the signal to the computer, and the computer gets the spectrum that we want.
Okay, so now we finish watch the video and we go back to our PowerPoint.
Jean-Fal, it's not very clear. Can you share again?
Yeah, okay, I'm trying to show
Yeah,
again.
now it's fine.播放模式。
Oh. So it's okay now?
Still not, still there's some areas like in dark. You stop sharing and then share again.
Sure again.
First stop sharing and then share again.
Okay. So this one.
Yeah, now it's fine.
It's okay.
Yeah, it's fine.
Okay, thanks. Okay, thanks a lot. Okay, we will continue our lectures, and we have shown the video for twice, and now is the Angelan the liquid equipment, and normally the HPLC named as the brand and model for such like this Angelan and the 1216 HPLC, and also we can see this the 5 mobile features bottles, and also is the pumper inside. And for this part, you can see is the automatical sample. And for this part is for the room temperature box. In here you can control the temperature in your separation and analysis process, and it's also the room also inside these boxes. And the last one in here is the detector. So okay, is another equipment in our library. Also is different models in here. For example, this is a mobile features bottles in here, and here is the automatical sample, and here is the pumper and the temper in here. And also can see here is the room temperature box, and also the detector will be listed in this part. So okay, we have prepared some questions for you, so you can answer them.
One minute for the poll.
Time is over.
Okay, thanks for your question answers about them. Okay. Dr. Yan, should we answer them this question?
Yes, you can go ahead and explain.
Yeah, okay. Okay. And for the question 1, of course, the sample mixture is separated at the column. The column is used to separate the sample mixtures. And for the question 2, here we can see the column length is generally in a range of normally we is 10 to 13 centimeters. Okay, for the third question, it's I see many answers for them, but for the HPLC, normally we to analysis the high boiling point micro and organical compounds is so the first answer. Of course, we can analysis all organical compounds for example for the analytical such as some proteins. It's very hard to use the liquid chromatography to analysis. Okay, finish these questions, then we will go to our lectures. And we will go to the third part. Is the usage of usage of HPLC. The arm of HPLC is qualitative and quantitative analysis. Also, before we start the lecture, we will see the video for the whole exams of using HPLC to do analysis work. Now this time I know how to show the voice. Okay. No.
Jean Fal, please go ahead. Continue your lecture.
Okay.
The suitable mobile phase was selected according to the experimental method. The mobile phase was configured. It was filtered through the 0.45 m filter membrane. Then, the bottle cap was loosened, and the ultrasonic was carried out for 10.15 mean to complete the preparation of the mobile phase. Pay attention to the mobile phase should be replaced in time to avoid bacterial growth affecting the HPLC. The samples were ultrasonically extracted with solvents. After standing for 2 hours, the precipitate was centrifuged and collected for polysaccharide determination. The supernatant was collected and appropriate methanol was added to complete the polysaccharide precipitation. The supernatant was centrifuged at 3000 rpm for 30 mean, filtered through 0.45 m nylon, and analyzed by HPLC. Open the HPLC analysis software and log into the chromatographic analysis interface. Open the exhaust valve, selecting the pump to be used. Adjust the proportion of the mobile phase to 100% and the flow rate to 4 milliliters/mean. Exhausting for 2-3 mean. After the exhausting, adjust the flow rate to 1 milliliters/mean and close the exhaust valve. Put the prepared sample into the sample rack and select the analysis method and the location of the sample, then click the single injection for sample detection. This is
Okay, after watching the videos, we have a simple understand how to operate the HPLC analysis. But for this, we will introduce to you the qualitative analysis of by using HPLC. Normally, this have 3 reference A, B, C, and we weight the right amount of standards and configures the appropriate concentration like this, and the samples will be injected into the HPLC analysis system. Then we will get the chromatogram for reference A, B, C as 3 steps. So this is for references and standards for the samples analysis. Normally the herbal samples normally will be placed in here, and suitable solvent, organic solvent will be added into this here, and the ultrasonical extracting normally be used, and after that we will centrifugans and filters to get the prepared samples, and these prepared samples will injected into the HPLC analysis. Normally for the HPLC analysis, we need to do is set up the chromatography method, and also the samples will be injected into the system. Then we will get this chromatogram for plant. This is using the liliesem as example. This is for the step 2. For the step 3, and we can get all the compounds, all the chromatographies together. Firstly, we can say this is the reference B in just under this calogram here, and also we have a reference C and the reference A together. And this calogram is the samples being analyzed, and we can find the same because they have the same retention time in here, and also the collector of these peaks are very similar. So normally this we have to do. By this way we can know this peaks is reference B and this peaks is reference C. And if you have any question about this, we can leave it here and we can answer it later. So for after the quantitative, this is quantitative analysis. We take the example of reference C as example because we can make them in different concentration, just like this, from a high concentration to a low concentration like this, and all these samples will inject into the HPLC analysis. And also we can see here, this is peaks, the areas, peak areas will increase the sample by sample with the concentration increased. So this is step 1 for concentration. After that, we can do is that this is reference A. We have its concentration, and also we can get the peak areas of this in this concentration, and just like this in here, this peak areas of this reference A in concentration 13.3, and also different concentration. They also have different peak areas, and the same for reference B and reference C. It has a different concentration and different peak areas. After we get this date, we will do the standard cover. So here we found the standard cover. We can find the relationships between these concentrations and the peak areas. Normally, the relationship are square. Normally, we are higher. The more it is equal to 1. That means the linearship has better. Okay, by doing so, we will can find is just we have said this is a string reference, and also this is the compounds in these samples. And for the peak C, we can do it integration. In using the integration tools in here, we can make sure the peak areas in the samples. For example, for the peak 1, the retention 10 is in 10.10 millimeter, and also the peak areas by using the integration is 1,214 and for the peak 2 in here, and we can say the retention 10 is 223.8, and the peak areas in here in the samples is 794, and also the same for the peak 3. The retention 10 is 13.3, and the peak areas is 5,869. But after get this useful information, after let's get this peak area information in the samples, we can substitute. Okay, you can say using this we can calculate the concentration of substance in the samples. For example, we get the here is the peak areas of compounds A, and this is the linear functions for it. So after we put them inside, we can get over concentration of compound A in the sample. Okay. And after that, we also can calculate the concentration according to the weight of the sample you have weighted. So this is the quantitative analysis of sample by using HPLC. Also, there have another questions for use.
You have 1 minute to complete this book.
Okay, thanks for your answer. And for the question 1, we the compounds that ensure that the mobile phases flows through the column at a stated rate are by using the infusion pump. The right answer is here, the infusion pump. And in Okay, this is maybe another question we didn't knowledge we didn't invite, and in liquid chromatography, in order to improve the analysis efficiency and shorten the analysis time, the driver must be used is graded at elution. Okay, the last part is the application of HPLC method. Because of the video program, so we will show some works we have done by using HPLC. The first application of HPLC of the application of HPLC. The first one is using to investigate the best harvest time for herbals. So for example, we harvest the herbals and we dry in the sunshine, and all the samples will be analyzed by using HPLC. And here we will get the HPLC fingerprint for different batches of herbals in here. And this is just one example. And here we can find about 20 common pigs in these batches of harvest herbal. And another important is that we can calculate the concentration of male compounds in these herbals, and we calculate it together by using the total concentration in here. And we can find in these pictures the best harvest time is maybe in the later, August, and also the same. The best time for in is because the different areas, the sunshine will be different, so we can show the best time for this in Sihu, in these areas, the different is in the middle of August. It maybe is the best time for harvest. This is one example. And the second example I want to show you is the process. As you can see, this is one herbal. It's the fresh one. After we process the one time, the color will be started from yellow and to the brown. And then if it's different process, the color of this of these herbals will be too dark, and it's very hard for you to identification how many times it will be it has been proposed. But here, just like this, 1, 2, 3, 4. After 4 times, the color all black, so it's very hard to identification the detailed times. But by using HPLC method in here, we can see the fresh one, and for the first 3 times, the peaks for in 6 and 8 in here, but we see higher than 5 times, the peaks will disappear. And also some as for the peaks 4 and 6, at when it's fresh with very low temperature, low concentration, and after 5 times processed, the concentration of peaks 4 and 6 will be increased.
Please little bit move your camera. We only can see your hair, not face.
Okay.
Now it's fine.
I'm fine, thank you. Just a little nervous about this. So by doing so, I use HPLC, we can help us to make sure the proper stance for these herbals. Okay. Is another example of how to use HPLC to do research, and this is of a plant named honey shaki, and this is the HPLC fingerprint of this herbal. And another thing is that we also call it the TCM preparations. For TCM preparations, normally it contains many different kinds of herbal together, and it's very hard to say if you forget to add the expensive herbals instead. But by HPLC, we can show in here, this is HPLC fingerprint. This is for this honey shaki, the HPLC fingerprint. They have many, it is filter compounds. And we can see here, if the preparations without these herbals, in here in these areas, we can't find the filter compounds in here. So that's by doing so, if it can help us to estimate the quality of preparations, and also to make to make sure it's fake or the real ones. Okay, here I want to also show you some research in all my group have been done. The first one is by using the HPLC. The first thing is that we using HPLC normally is UPLC Archer. The speed is much faster than the common HPLC, and we have been analyzed 11 compounds in here in 9 minutes. You can see this is HPLC chromatography, and we have been analyzed 11 compounds in 9 minutes. And another thing is that we can help to classification different types of products. Okay, because we different products with concentration plus custom. The last example I want to show you is our group's new publications. The application of using the deep electrical solvent. And we normally know if you want to extract the herbals, normally or the organical solvent will be used. But in here we using the new synthetic deep electrical solvent as extractor being added in here. After that, we have to find that is much better than the traditional or conventional organic solvents. And this our artist has been just been accepted publication in analytical method as a hot artist being recorded in this way. And also this is a cover. We have been invited to as a cover artist being publication in analytical method. In here, the advantage of the green deep elucidator solvent is firstly is a green solvent. It can be by this graded, and also it can replace the usage of conventional organic chemistry. So we said is a green extracting method, and the extracting effect is much better than the conventional ones. Okay, the fourth part is the supporting information. There's some information about in our lectures. If you don't understand it, we can see our PowerPoint, and also this PowerPoint and videos we will show with you. Here is the preparation of water filters, and is also showed in our videos. Okay. Okay, this is my group now, and thanks for them to prepare the material and the purpose of this PowerPoint. This is my students. And also, this thanks very much for Professor Igbo and for SCBS to invite me to join these lectures. Through some, there are some issues in the video part, but also thanks for you. This is our team in Changsha. And also, if you have any question, you can come to Changsha to find me. And also, you can have send me email about this. Thank you very much for your listening.
Okay, thank you so much, Dr. Jan. This is a very informative lecture. So today's session is now end. And if there is any question, please ask.
So you can come to Changsha or the.
If there is no questions, or if you can write the question, we will answer in a chat box.
Sure, you can send me a message, and we also have a discussion tomorrow.
So tomorrow at same time, 12 noon at the time of Pakistan. So we will start the remaining presentation of Okay, sorry, we forgot we have a last poll. So before closing this session, we want to just inform you this, just fill this poll to just for your learning and for better understanding for us to how much you learn from this presentation. Please. Okay, thank you so much. Now poll is completed, and now there is a good results. The most majority gave the right answer on that poll. So if there is any more question regarding this today's presentation, kindly send by the in the chat box, and we will happy to answer about your questions, about your queries. And tomorrow at same time, Pakistani time, 12 noon, we will start the remaining presentation. Tomorrow, from my side, I try to explain more practical side, and from Dr. Yan will also presenting on the semi-preparative HPLC, which is the she will explain tomorrow at what time, Dr. Yan? At 1 o'clock? 1:30. So Dr. Yan will deliver her lecture at 1:30 Pakistani time, and inshallah I will start at 12 noon. So thank you so much. For Dr. Jinko, you have any comments on it?
Oh, thanks. Don't have more questions. See you tomorrow.
Jean Fal, is it possible to share your slide to the participants? They are asking.
Yeah, of course. You can show all the information from PowerPoint and the videos also.
Thank you so much.
Okay, thank you so much. And we will start tomorrow at same 12 noon. And till that, goodbye. And we will see you tomorrow. And Dr. John, we are sharing the presentation as well on your email ID or